All right, let's get straight into it. If you're tackling your GCSE biology revision, you know these required practicals are a big deal. Today, we're going to break down required practical number 8, all about plant growth. Let's make this super clear.
So first up, let's start with something you've probably seen 1,000 times. You get a houseplant, you stick it on a shelf, and within a week or so, it's doing this dramatic lean towards the window. Why? What's going on there? Well, it's not just a random thing. It's a really clever survival strategy, and it's the key to understanding this whole practical.
And that bending action, it's all controlled by something called a tropism. You can think of tropisms as the secret rule book that plants follow, telling them how to move and grow to get exactly what they need from the world around them.
So what even is a tropism? In simple terms, it's just how a plant grows, either towards or away from something in its environment, a stimulus. It's like a built-in instruction. If you sense light over there, grow in that direction.
And for your GCSEs, there are two big ones you need to know. First, you've got phototropism. Photo means light, so this is all about growing in response to light. The shoots, the leafy bits, grow towards the light to soak up as much energy as possible for photosynthesis. Makes sense, right? Then you have gravitotropism, which is all about responding to gravity. Shoots grow up, away from gravity, reaching for the sky, but the roots do the opposite. They grow down, towards the pull of gravity to anchor the plant and find water.
So, how does the plant actually manage all this? It's not thinking about it. It's all down to chemistry. The real hero of the story is a powerful little chemical messenger, a plant hormone called auxin. This is what tells all the cells what to do.
Now this is the really cool part about auxin. It controls growth by making cells get longer, a process we call cell elongation. But here's the twist. It works differently depending on where it is in the plant. In the shoots, lots of auxin means lots of growth. But in the roots, a high concentration of auxin actually stops the cells from growing. This difference is absolutely crucial to remember.
Okay, so we've got the theory down. Now for the main event. Required practical activity 8. This is where you get to be the scientist and gather the evidence for everything we've just talked about.
This is the central question your experiment needs to answer. We're not just going to watch some plants bend. We are going to design a proper scientific experiment to prove that it's the direction of light that's causing it.
So here's the game plan. You need two groups of seedlings. Your experimental group gets light coming from just one side, and then you have your control group, which gets light from all around. Now, this is important. Everything else has to be kept exactly the same for both setups. Same amount of water, same temperature, all those things are your control variables.
So why do we even need that second group with light all around? Well, that's your control, and it is so important. The control group acts as your baseline. It proves that the bending you see in the first box is definitely happening because of the one-sided light, and not just because cress seedlings like to grow sideways. Without a control, your experiment isn't really a valid scientific test.
Okay, the experiment is all set up and running. Now for the bit that actually earns you marks in an exam, recording your results like a pro.
You're basically a detective gathering 3 types of evidence here. First, the numbers, the quantitative data. You'll need to measure the height of your seedlings. Second, the visual evidence. This means making big, clear, labeled scientific drawings. And third, your qualitative observations. Just writing down what you see, things like which way they're growing or if their color changes.
And when it comes to your numbers, a nice, tidy table like this is the perfect way to present them. It's so clear, easy to read, and it lets anyone, like an examiner, immediately compare what happened in your experimental group with what happened in your control group.
And a quick pro tip on those drawings, because this is where people lose easy marks. Don't sketch. Use a sharp pencil, draw single, clean lines. Make your drawing nice and big, at least half a page, and use a ruler for your label lines. It just screams, I know what I'm doing.
Okay, let's connect all this directly to what you'll see in an exam. The examiners don't just want you to know the facts, they want to see that you can think like a scientist.
You can pretty much guarantee you will be asked about control variables. Remember, these are all the things you have to keep the same to make sure it's a fair test. Things like the number of seedlings, the species, the temperature, the amount of water, you get the idea. The only thing you want to be different between your two groups is the direction the light is coming from.
So what should your results look like in the end? Well, if you were to calculate an average and plot it on a bar chart, you'd expect to see something like this: a really obvious bend, or high curvature, for the seedlings with one-sided light, and pretty much no bending at all for your control group. A chart like this makes your conclusion impossible to argue with.
And as you're doing all this, the measuring, the observing, the drawing, you're actually demonstrating a bunch of key skills. In the spec, they're called AT skills, for apparatus and techniques. You're showing you can measure accurately, observe changes carefully, handle living things correctly, and make proper scientific drawings. It's all part of what's being assessed. So, we have to bring it all together. This is the big takeaway. In your seedlings, with light from one side, the auxin hormone moves over to the shady side of the shoot. That buildup of auxin makes the cells on the shady side grow longer and faster than the cells on the light side. And that unequal growth is what physically pushes the shoot and makes it bend towards the light. Your results are the evidence that proves this whole theory works.
And finally, let's just zoom out for a second. Understanding how to control plant growth isn't just about passing an exam. Think about it. If we can manipulate how plants grow using light and hormones, what could that mean for the future? Could we design super efficient vertical farms right in the middle of our cities? Could we figure out how to grow food on another planet, like Mars? It all starts with the basic principles you've just proven and this very practical. Pretty cool, huh?